This is Selma Schimmel in San Antonio at the annual San Antonio Breast Cancer Symposium, where we're joined by Dr. Cliff Hudis, is the Chief of Breast Cancer Medicine Service at Memorial Sloan-Kettering Cancer Center in New York City. Hello, Dr. Hudis.
Hi.
Would you take a few minutes out to give us your perspective on some of the issues and challenges that you deal with on behalf of biospecimen, tissue collection, tissue exchange, and exchanging that information?
So the issue of the massive amount of data that you can now generate from a single patient, a single specimen, in terms of genomics, and how that gets linked to that patient's clinical trial results or even conventional therapy results is a big component of what we want to accomplish with, again, CancerLink. So in this case, you could imagine that you have now thousands upon thousands of patients, and 3% of them have a given genetic abnormality, which any individual clinician isn't going to be able to pick up necessarily as important. But a rapid learning system could have the potential to identify that people with a particular variation always have a particular toxicity or frequently seem to have a higher response rate or whatever. And I think that the promise right now is really there. And we see it in presentations at this meeting in ASCO, where we see narrowly targeted drugs in narrowly selected patient populations with benefit. But it would be interesting if we could find some of those things out sooner because of what we're able to glean from these rapidly developing databases.
On the subject of all of this genomic profiling, I'd like to get your opinion on whether you feel the whole concept of tumor grafting, and in this case mouse grafting, which I've been reading companies are doing this, how viable is this, where you will get the surgical specimen and it's a large enough specimen very quickly, and it is transplanted into this mouse, and the mouse obviously then grows the cancer, and then they try to treat the mouse. How real is this for the future?
I don't know yet, because obviously in many circumstances, mouse models of illness work. In many circumstances, they either don't predict what we have in the human, or their predictions aren't in the same magnitude, and so they end up misleading that way. So we'll have to see. One of the things that we do have to keep in mind, though, and this is, goes back to an earlier comment you made, is that the human is a big organism with a cancer. But that organism metabolizes drugs, eats certain foods, engages in certain behaviors, is exposed to certain environmental influences, all of which isn't to bring in voodoo, not at all. Those things interact ultimately with your genetic base and lead to changes, of course, in terms of how drugs are cleared and how they work. So on the one hand, for a small monoclonal tumor, the kind of system you described might work. But if you haven't sampled all of the cancer, if the part of the cancer that's micrometastatic and unseen has an evolved genetic background compared to the tumor you do get, there are a fair number of potential pitfalls with such a system.
How do we protect patients right now with this numerous companies that are out there that are available? You can send for molecular profiling. Here's a molecular pathology company, or we're a genomic profiling company. How is a patient to know? And these are costly analyses. Where to go? You know, how do you separate hype from fact?
Well, we walk a fine line because many of us share the enthusiasm that these kinds of analyses will ultimately be important in improving patient care, either because of selecting patients in the first place or because they'll allow us to predict the impact of a therapy while a patient's on it. So we have to see them developed. And some of them have gotten further along than others, but I think it's fair for a patient to ask, considering this test, what is the concrete evidence that I will be able to use this result to make a better treatment decision right now? At the same time, again, if we were doing this on a broad scale, even today not knowing how to use it, but we were gathering the information in a very regimented way, we might in a year or 2 or 5 start to associate some of the currently observed changes with outcomes and start to maybe at least test those associations, ultimately leading to the utility of these of these very expensive sometimes tests. So I think it's a fine line about when a test should be done or not. But I will go back to my earlier point, which is for a patient, especially paying for a test like this, I think it is very fair to ask, Doc, how will you use this to improve my care? And if there's a clear-cut answer, then maybe the test should be considered.
Do you think there is a place now for patients at the time of their surgery to have a discussion with their doctor saying, hey, I would also like my tissue, if you don't do it at this institution, I want it to be sent out for molecular pathology and a genomic profiling for my cancer type?
So it's entirely dependent on the cancer and the stage and the therapeutic options that follow. I have some patients right now with far advanced breast cancer who will, of course, ask about this because there's a lot of hype, but they've actually been through many of the available therapies, and there's been no clear evidence that such a test for those people could make a difference. And that's frustrating and disappointing given all the hype and enthusiasm for it. On the other hand, in breast cancer, going back a generation or more, we've done a simple version of this always, right? We always test for estrogen receptor, progesterone receptor, and more recently HER2. Why do we do that? We do that because those molecular targets lead very directly to changes in therapy and better outcomes. And I don't think that standard's changed. If we move up a level, and now we're talking about doing it at the level of the DNA or the RNA, those are important to do as well in some circumstances. But I think it's very, very fair to ask the simple question, if I do this test, is it going to be useful? And let me just caution everybody one other way. Some people will say, well, I hear you, but I want to do it now so I have it in the bank for if I need it later. The problem with that at the moment is that the rate of evolution in the technology is so rapid that you might want to consider an alternative approach. For example, freezing material at the time of surgery and not processing it just yet.
Great idea.
Because maybe the test you want to do in 2018 is 3 generations beyond anything being done today, and you don't want to waste your tissue and your money prematurely.
That's a fabulous idea. And where does the tissue get stored? There are companies that will cryo tissue companies?
You know, there are multiple models for this, and there are different pathologic approaches. In the United States, most of the time tissue, at least in breast cancer, is embedded in wax, paraffin. But in Europe, freezing the material has been more common. So I think that this is an issue for us to discuss.
Thank you so much, really. Dr. Cliff Hudis, Chief Breast Cancer Medicine Service at Memorial Sloan-Kettering Cancer Center, New York City.
Thank you, you too.